Hi, good afternoon. My name is Naomi Dillard, and I am a junior biology major at Prairie View A&M University. This last summer, I had the opportunity to do a summer research internship at the Texas A&M College of Medicine in the Department of Physiology under Dr. Joseph Rakowski. So I will share the poster. Okay, so the title of the poster that I worked on was Genetically Augmented Renal Lymphangiogenesis Alters the Immune Response Following Acute Kidney Injury. And my specific area of interest was the specific immune cell subpopulations that are present after inducing lymphangiogenesis and injury. So first of all, what is acute kidney injury? It can be characterized as a sudden decline in renal function that causes a buildup of waste products in the blood. And if untreated, it will eventually progress to chronic kidney disease and end-stage renal disease. So we wanted to know if increasing the amount of lymphatic vessels in the kidney would decrease the amount of cases that eventually progress to chronic kidney disease. The lymphatic system is a network of vessels that regulate fluid and solute homeostasis. They also participate in immune cell modulation and differentiation. Now, lymphangiogenesis is the growth of new lymphatic vessels and also the dilation of existing vessels. And it is promoted by the binding of the VEGF-D protein to the VEGFR3 signaling receptor. The lymphatic system is important and plays a big role in injury resolution because they also play a role in immune cell trafficking. So as described down here, at the onset of injury, pro-inflammatory cells are recruited and needed for healing, but anti-inflammatory cells like T regulatory cells are needed to balance out this response. And without this balance, subpopulations of cells like Th1 cells and Th17 cells will cause continuous tissue damage, eventually leading to fibrosis and chronic kidney disease. So to better understand the impact of lymphatics in resolving inflammation, we used a transgenic mice model termed the KidVid model up here, which is based on a TET-on system that overexpresses VEGF-D in the kidney. And VEGF-D is what promotes lymphangiogenesis. So using this model in a previous study, the Rakowski lab compared KidVid positive mice, which is here, with wild type mice, and found that the mice with increased lymphatics exhibited reduced fibrosis and a restored glomerular filtration rate. And this led us to hypothesize that increased lymphangiogenesis would modulate immune cell subpopulations and thus regulate inflammatory responses during injury. So to test this theory, we looked at two models of injury. We looked at the podocyte ablation model and the cisplatin model. And in both models, we induced lymphangiogenesis by giving the mice doxycycline in their water. And doxycycline has been found to activate the VEGF-D, which promotes lymphangiogenesis. So we induced lymphangiogenesis for 21 days. And then on the 21st day, we induced the injury. So that starts like day one. So day one, off the water, we induced injury. And then 7 days after injury, we killed the mice and harvested the kidneys and the spleens to use flow cytometry to analyze their immune cell subpopulations that are present. So a little bit about flow cytometry. It's essentially hydrodynamic focusing that allows cells to pass through one at a time as lasers pass through them. And we can measure the light scattered from the cells to give us an idea of how big the cell is, and the fluorescence emitted to tell us which labeled antibody was attached to it. And this is an example. So this is an example of a plate that we use to feed into the machine. So we would have like kidneys here, spleens here, and liver here. And then in each row, we would put different antibodies that we would then look for. And this is an example of results that we would get from the flow cytometry machine. So this is how we gated. We would first gate for cells that we believed to, like the things that we believed to be cells. And then we would look for live cells, and then we would look for the different antibodies. So our results showed that there was a decrease in the overall number of pro-inflammatory cells and also an increase in T regulatory cells in our resident tissues. In our restimulated tissues, we had a lot of cell loss, so we can't be completely confident in these results. But for our CD8 cells, we didn't see a decrease in pro-inflammatory cytokines, but in our CD4 cells, we did see an increase in T regulatory cells. So, yep, we saw a decrease in the Th1 and Th17 response in mice with increased lymphangiogenesis and an increase in T regulatory cells. And this leads us to believe that enhanced lymphatics does help to reduce the pro-inflammatory response and modulate the inflammation with Tregs, preventing the progression to chronic kidney disease. And our next step would be to increase the number of animals used in the study so that we can actually run statistics on our data and get a more accurate depiction of what's really going on. So acknowledgements, I was supported by a fellowship from the NIH, and I'd also like to acknowledge Gaurav Barnwal, who was the postdoc that I worked with, and Dr. Joseph Rakowski. So thank you for listening to my presentation today. I hope you learned a lot. And if you have any questions, please don't hesitate to ask. Thank you.